Well, greetings to everyone, and welcome to the Science of Childhood Cancer lecture series hosted by the St. Jude Comprehensive Cancer Center. My name is Greg Armstrong, and on behalf of Melissa Hudson, who along with me is a co-leader of the Cancer Control and Survivorship Program within the St. Jude Comprehensive Cancer Center, I welcome you to this lecture. It is my great pleasure today to welcome our speaker, Yutaka Yasui, who is my colleague in the Department of Epidemiology and Cancer Control here at St. Jude and a member of the Cancer Control and Survivorship Program. Before I formally introduce Dr. Yasui, just a note that you can submit questions at any point during this talk using the chat function and sending your questions to all panelists. If there are additional questions, Dr. Yasui has kindly offered to stay on for some time after the top of the hour for anyone who'd like to have further discussion. And now I'm delighted to introduce to you Dr. Yasui. Yutaka received his PhD in biostatistics at Johns Hopkins University in 1994 and worked at the Fred Hutchinson Cancer Center and the University of Alberta for 10 years before joining St. Jude's Department of Epidemiology and Cancer Control in 2015. His research focuses on population science methodological issues, particularly those associated with childhood cancer survivorship, risk prediction, and data-driven biological investigations. He is currently a principal investigator of 4 NCI-funded R01 studies and a co-investigator on numerous additional NIH studies, including the St. Jude Lifetime Cohort and the Childhood Cancer Survivor Study, or CCSS. And speaking of CCSS, Yutaka was the lead biostatistician at the founding of the study in 1994 and currently serves as the chair of the Epidemiology and Biostatistics Working Group for CCSS. He's been the author of over 450 peer-reviewed publications and has an H-index of 115. And over the last 20 years, he has developed statistical methods for multi-locus interaction and haplotype analysis in genetics, gene set pathway analyses of transcriptomics, and mass spec-based cancer biomarker discovery in proteomics. And then most importantly, he's been the recipient of several teaching awards and mentoring awards through the years. Most recently, in 2020, he was named the St. Jude Mentor of the Year. So Yutaka, welcome. Thank you for accepting to give this lecture, and the stage is yours.
Great. Thank you very much, Greg, for a nice introduction, and Dr. Roberts and Dr. Mulligan for having me here for to speak today. So today I'd like to start with this intriguing observation from Nordic twin study of cancer, which is a population-based study, high-quality study involving more than 100,000 female twins. In this study, they found that monozygotic twins and dizygotic twins had the same lifetime breast cancer risk. And also, when both twins had breast cancer, the average interval between the two cancers was the same. However, when one twin had breast cancer, the other twin's rate of breast cancer differed. For monozygotic twins, it was 1% per year. For dizygotic twins, that was 0.5% per year. So this is a little bit strange because conditional rate of the other twin is half after one twin had cancer, and yet the interval between the two cancers when they both had it is exactly the same. That is like one has a sports car and the other has Toyota Corolla, which going half the speed, and yet it takes the same time to arrive to the destination. Now, for some people, this is very natural and totally makes sense. But if you follow the current dominant paradigm of genetic association studies, this is strange. So I'm going to come back to this later. So today I'm going to talk about these 3 topics, which is all related to what we can learn from genome-wide association studies. And I'm interested in this because we are engaged in childhood cancer survivorship research, and genetics is highly relevant for predicting risk of late effects. So before going to jump on the genetics, I'd like to talk a little bit about survivorship research. So as many of you know, 5-year relative survival of childhood cancer patients has increased from 20% in 1950s to over 85% today. Because of that, we have more than 500,000 child cancer survivors in the US alone. And every year, we have additional 12,500 new cases becoming 5-year survivors. Now, last year, we looked at excess deaths led by Annalyn Williams of our department postdoc. And excess death is a death counts over and beyond what is expected for age and sex matched US general population. So if the cancer patients die the same way as age and sex matched US population, how many more deaths we are observing compared to that expected? And that number is declining if you look at less than 5 years from diagnosis. Especially when you look at acute lymphoblastic leukemia, in all three groups of less than 5, 5 to 9, and 10 plus years from diagnosis, in spite of the increase in the cumulative number of 10 plus year survivors every year, the total count of excess deaths is declining over years. However, that's not the case for every single diagnosis. Hodgkin lymphoma, for example, less than 5 years and 5 to 9 years group are doing pretty well. But because of the increase of the survivors' total population who are exceeding 10 plus years every year, the excess death number is also increasing linearly. And if you look at all childhood cancer survivors total, then excess death counts per year has not declined as much as those within 5 years. And that is because of this red group, which is 10 plus years, gradually increasing the number of excess deaths every year. So even though among the total excess deaths, less than 5 years is still the majority, the increase over years of the 10 plus, 10 plus years from diagnosis group is concerning. And that strongly motivates us and shows a pressing need to study survivorship after 5 years of childhood cancer diagnosis. Now, these survivors experience, as Nikhil Bhatta reported several years ago, 17.1 chronic health conditions by age 50, and 4.7 of which are severe, disabling, life-threatening conditions or death. At St. Jude, we tackle the survivorship issues led by Les Robinson and Melissa Hudson across clinical, population, biological, and data sciences. We work together like a giant one laboratory and tackle different aspects of the survivorship. And we have received this Team Science Award from AACR in 2019. We are very privileged and fortunate to have exceptional resources to study survivorship. As Greg mentioned, childhood cancer survivorship study is a 32 institutions collaboration in North America. St. Jude Lifetime Cohort Study, graduate of St. Jude, and outcomes rate effects are clinically assessed. And both studies have genetic materials and information, led by Les Robinson, Melissa Hudson, Kiri Ness, and Greg Armstrong. Now we are sharing this data under the leadership of Jinwei Tsang in computational biology through St. Jude Cloud. And in particular for cancer survivorship portal, Shinzo and his team in computational biology in collaboration with us is adding analytic capability to both the clinical data and the genetics data. Today, I'm fortunately with the time limitation, I cannot go through this resource, but if you have a chance, please visit St. Jude Cloud on our internet. Now, genetics. Now, Richard Lewington is a prominent geneticist who wrote this 1972 paper called The Apportionment of Human Diversity. In that paper, he studied these many populations across the world, categorized by so-called races. What he did is he looked at all these genes and looked at their diversity across individuals and decomposed that variation into between races, within races, between population, and within population. This is called analysis of variance in statistics. And he found that the majority, by far the majority of the variation exists within populations, not between races or between populations. He said, the results are quite remarkable. The mean proportion of the total diversity that is contained within populations is 85.4%. Less than 15% of the all human genetic diversity is accounted for by differences between human groups. Based on randomly chosen genetic differences, human races and populations are remarkably similar to each other, with the largest part of, by far, of human variation being accounted for by the differences between individuals. Now, his finding has been repeatedly confirmed, and public education, such as biological science curriculum by NIH, uses that information and shows the variation picture in this way. Also, nature education cites the 85%, 15% split in their discussion of human evolutionary tree. However, today, that message seems a little bit odd when we see 23andMe and Ancestry DNA offering a service that detects people's ancestry and geographical origin. In fact, 2008 Nature paper, these researchers showed that using individual DNA, you can infer their geographic origin with surprising accuracy, often to within a few hundred kilometers. So what they show here is that the two-dimensional space created by the genetic variables PC1 and PC2, which is principal component, direction of the greatest variation in genotypes, that is PC1, and perpendicular to that direction, the second greatest variation in the genotype is PC2. If you plot each individual along these lines, then you can separate small group of Europeans as shown here. And prominent statistician geneticist Anthony Edwards wrote a paper in 2003 talking about Lewinton's fallacy. So was Lewinton's 85%, 15% split genetic variation wrong? It seems like Edwards is implicating that. Now, statistically, this can be understood as univariate and multivariate statistics. So I'll illustrate this concept. So here you have marker A and B to try to distinguish population 1 and population 2 in a two-dimensional space. If you use A alone, univariately, which is to look at marker A's value to go this direction, then you see the population 1 and population 2 are strongly overlapping, cannot distinguish the two populations. Similarly, B alone cannot distinguish the two populations. However, if you look at this two-dimensional space multivariately, A and B together can distinguish the two populations completely. Along this direction, left is population 1, right is population 2. So this analysis was looking at the genotypes of SNPs, single nucleotide polymorphisms, multivariately and deriving PC1 and PC2 with a multivariate analysis. The same first author wrote another paper showing that if you use only 100 SNPs, then you cannot see the patterns. As you increase from 100 to 1,000, 10,000, and 100,000 SNPs, then the detailed picture begin to emerge, and you need a lot of loci SNPs to distinguish different populations. Now, principal component takes this form. It's weighted sum of SNP alleles. That is the number of minor alleles at each SNP multiplied by its weight. So betas are the weights. You multiply the number of minor alleles that each individual carries at each SNP site, and then you add it up. That's principal component. So what we can learn from this story of Lewington is that looking at each single SNP at a time, you cannot find patterns. Looking at multiple SNPs jointly, then patterns may emerge depending on how many SNPs you are using. So was Lewington wrong? Not necessarily, because he said based on randomly chosen genetic differences, which means take one SNP randomly, then it is a situation where you are looking at each single SNP and you cannot separate the population. So what he said is not technically wrong. And his analysis approach is not surprising because there's a long tradition of looking at single variants one at a time in genetics. And it's been very successful for Mendelian diseases. So Mendelian diseases are caused by single mutations. And this paper in 2019 reports that gene discovered for Mendelian conditions and the new Mendelian conditions themselves have been increasing since the development of positional cloning in 1955. And recently, that technique has been expanded with next generation sequencing, and the discovery speed of the Mendelian diseases continue to go up. So polygenic risk score, is this a univariate or multivariate look of the genes? It is a cutting-edge method in genetic risk prediction for precision medicine, and even newspaper talked about it several years ago. So this paper, this article from New York Times says, with a sophisticated new algorithm, scientists have found a way to forecast an individual's risks for 5 deadly diseases. And the article is referring to this paper in Nature Genetics, which is written by Boston Group, who says, for most common diseases, polygenic inheritance involving many common genetic variants of small effect plays a greater role than rare monogenic mutations. And they looked at coronary artery disease, type 2 diabetes, breast cancer, etc., using 6.6 million or more variants. Except the case of breast cancer, they looked at 5,000 SNPs. With that 5,000 SNPs, they can differentiate the top 1% of the polygenic risk score carriers compared to remaining 99% by a risk ratio of 3.36. And as this graph shows, that the percentiles of polygenic score differentiates people's risk smoothly and continuously using this score. So the idea of this polygenic risk score is something like this. There's a population of women. Some carries SNPs 1, SNP 2, SNP 3. Each makes a risk of breast cancer a little bit higher. And because you use all the 6.6 or 5,000 SNPs, you are going to differentiate different shades of risk. So overlapping area has a higher risk, and non-overlapping area will have a lower risk. And because of the number of SNPs you enter to the polygenic risk score, you have a smooth risk gradient over the whole range. Now, polygenic risk score takes the same form as principal components. It is number of risk alleles at each SNP times the weight, where weight is the effect of the SNP allele on the disease. So it is the same weighted sum of SNP allele format as principal components. But they have 6.6 million weights to estimate. How do they estimate these betas? Before we go to that issue, I want to talk about what SNP is really in our equation. So after completion of the human genome study project, HapMap project in 2005 published a paper about how haplotype variation exists across the human population around the world. Now, haplotype is a collection of alleles that are inherited from parents to offsprings through the inheritance on the same chromosome. So here we have 7 haplotypes, and there are a bunch of SNPs on each haplotype. But in order to distinguish these 7 haplotypes, they say all we need is a tag SNP that are colored here with different colors. So if you type these 6 different colors, one from each color, then you can distinguish those 7 haplotypes. So originally, SNP was motivated to tag haplotypes, which is natural because haplotype is the sequence that is eventually translated into the polypeptides that are biologically functional. So looking at haplotype makes sense, but that can be tagged by SNPs. So this picture kind of illustrates a key point where each SNP actually has only 2 types of alleles, minor or major, or referent and alternative. No matter how you label it, there are usually 2 types of alleles. However, there are multiple haplotypes in the same region because different SNPs tag the different haplotype. So here SNP123 tags the 3 distinct haplotypes. If you analyze one SNP at a time, you're comparing one haplotype compared to all the others. That is a little bit problematic unless that haplotype, let's say red, is a risk haplotype, and none of the others are risk haplotype. If 2 of the haplotypes, let's say red and the blue, are risk haplotypes, then if you compare red and all the others, analyzing one SNP at a time, then blue haplotype is in the other group and elevating that group's risk. So red versus others contrast may not reveal the red as a risk haplotype. So similarly, in these 3 SNPs, it's not tagging the specific SNP each of the 3 SNPs. You have to look at all these 3 together to distinguish these haplotypes. So again, looking at multivariately is needed to distinguish and understand each haplotype's risk contribution. This is a plot of the SNPs according to their frequency and chromosomal location across 6 populations, 2 Africans, 2 Europeans, and 2 East Asians. And for European samples, I plot the recombination rate from Iceland population. So if you see the horizontal line, that means alleles are correlated. They are on the same haplotype, and they are segregating together. You can see that these horizontal lines form a block across all populations. So for example, this orange is a block and this gray is a block. And between the block, there's a high recombination rate area. So the haplotypes in each block is going to be shuffled and connected to another haplotype in the different blocks. This plot is showing the haplotype of length greater than 100 kb, 100,000 base pairs. If you plot lengths greater than 1,000 base pairs, it covers majority or almost all of the chromosomal regions. So this is not anything special for chromosome 1. If you plot this for all the across the genome, you will see haplotypes everywhere. And this is the reason why we can tag with smaller number of SNPs than the exact number of SNPs exist in the whole genome. So how could we estimate the 6.6 million weights from a study of much less than million people? The technique they use is developed in 70s, and this particular paper, Efron and Morris in 75, talks about how to use it. They use it to estimate baseball players' seasonal batting average. They first observe beginning of the season batting average. So Clemente is hitting 40%, Alvis is hitting about 15% at the beginning of the season. And over the season, the batting average will be getting closer and closer to about 25%. That's what average hitter would hit in the season. So there are two information here. That is each individual's initial, let's say, first month's batting average. That is a good estimate for each person. However, another source of information is if you consider all batters as a human being playing baseball, hitting the ball, then 25%, or in this case, 26.5% across all players in the first month may be also a good estimate. So if you balance these two estimates, you're going to shrink the estimate towards the center, and that is called empirical Bayes estimate or James Stein estimates. And this idea is to borrow strengths from other batters to estimate each individual player's rate. And essentially, it is shrinking the estimate towards the population mean of the batters. So the same technique has been used for healthcare provider profiling. So Morris's student Cindy Christiansen in 1997 published a paper how to rank hospitals for something like surgical mortality, adjusting for case mix differences across hospitals. And the same technique is used even today by Agency for Healthcare Research and Quality to estimate each hospital's quality of care and trying to improve some of the lowest indicator hospitals. The same techniques is also used to estimate small area mortality and disease incidence. We applied that technique to Spanish Atlas of Mortality that I was involved in 1990s. And this kind of plot is helpful for thinking about hypotheses for etiology and also informing policy decisions. So mathematically, the method to estimate those 6.6 million weight is to fit a standard model for each SNP, but consider that effect beta to follow a certain distribution with mean zero and some variance function. Now, this thinking about all the betas to have come from a single distribution allows borrowing strength idea. So this is the key in the Bayesian hierarchical model to consider the effect to be coming from a single population. So in the case of batters, we are talking about population of all batters. In the case of SNPs, we are talking about population of all SNPs. Now, PHS is derived actually doing this way. So in some sense, you're looking at the univariate, each SNP univariately for SNP1, SNP2, and up to SNP 6.6 million. And then you're shrinking all estimate towards the population average and summing up. Now, that is not the same as looking at 6.6 million variables at the same time multivariately. So it is not a multivariate approach. The key question in this technique is from which population which we should borrow strength and in what form of variance we should use. So if you borrow strength from all batters in the left versus you stratify the players into pitchers and non-pitchers, and pitchers don't usually hit as well as non-pitchers, then you will do better. Your estimate will be better. Except some outliers like this who pitches and hits at the same time. Then it's a little bit screwed up, but in general, this strategy should work. So the most recent paper that was published last month does that for SNPs. So they consider, and other people do that too, but they summarize here that if you consider SNPs in the conserved region, coding region, UTR, 5' and 3' and transcription starting sites, etc., these are the groups of SNPs that carries higher effect in general over a variety of phenotypes. And therefore, if you stratify by these functional annotation of SNPs, you will do better for estimating SNP effects. So over the last 10 years or so, there are a number of Nature Genetics papers talking about how to form this different population of SNPs by frequency, linkage disequilibrium, which is neighborhood correlation of the SNP alleles, and also functional annotation that I talked about. So the same paper that was published last month looks at the variety of phenotype and decomposes the portion of heritability, that is inherited risk, due to SNPs that are in the polygenic risk score. And the red part is the part that is not explained by SNP, not by the polygenic risk score. So if you look at hypertension or BMI about 70% heritable, half of the heritability is still remaining to be discovered, but half is captured by polygenic risk score, and the same for the hypertension. So in our survivorship research, we utilize these results. So Dr. Zhao Min Wang of our department wrote a paper in 2018 utilizing at that time the cutting edge PRS of 170 loci for breast cancer that was published in Nature the year before. And he found that if you divide the PRS group into quintiles, so 20%, the top 20% women have 2.7 times higher risk of breast cancer than the lowest 20%. Now, this contrasts with another variable he created, which is mutations, known mutations that are known to cause breast cancer. And if you count these, he found that 4 out of 47 breast cancer cases he has in his data are from the carriers of one of these mutations. And if you calculate the relative risk for that, it's actually much higher, 21.8. But it's a rare mutation, so rare variant, so it is a small number of people who carries this risk factor, which is about 3% of our survivors. Dr. Sapkota in our department used hypertension PRS and obtained about the same gradient of risk, 2.7%, using Nature Genetics 2019 papers PRS for hypertension. He also calculated how much is due to genetics and how much is due to cancer therapy in the case of hypertension. And he found that 21% is actually explained, of the risk is explained by polygenic risk score and 15.7% by cancer therapy alone. So in this particular late effect outcome, the genetic risk is not the ignorable component for the risk prediction. He also has an impressive paper on coronary artery disease, and as shown here on the left, without PRS using only the clinical variables, he can separate pretty well. But with PRS, the high-risk group is further enhanced for their risk prediction. Dr. Cindy Im of University of Alberta has looked at transferability of PRS and GWAS hits. So general population GWAS generates PRS and GWAS hits, but is it usable in clinical population, such as childhood cancer survivors? Her conclusion is that it depends on the phenotype. Some phenotypes are influenced by treatment greatly, in which case less likely to generalizable from the general population to clinical population. So therefore, she and her colleague Dr. Yang Yuan at the University of Alberta are developing survivor-specific PRSs involving gene treatment interaction through NCI-funded R21. So some comments about polygenic risk scores. PRS with millions of SNP alleles can predict disease risk of each individual with some accuracy and precision. Is it usable in practice? That depends on the cost benefits as well as frequency of true and false positive and negatives. Because imperfect prediction, which is always the case, we don't have the perfect prediction. Imperfect prediction is subject to consequences. So we need to evaluate those before implementing in practice. Can the current PRS be improved for prediction? That is the theme that we have been working on. Sorry. And we have three thoughts how to improve it. One is to use a model that is based on biology or hypothesis on the truth. Second is what I have been talking about, multivariate approaches instead of sum of shrunken univariate estimates. And many people might say, why don't you use AI machine learning? But in order to use such flexible models, you need a massive data. If you think about where AI and machine learning are successful, it is games, automatic driving, face recognition. There are massive amount of data available and they're continuously collecting those data. So model can be quite detailed and flexible. In medicine, that may not be the case depending on the problem. So we take the first approach, and I'm going to report that approach result here. That is this hypothesis of genetic heterogeneity and rare mutations. So Mary Claire King, who found the BRCA1 locus in 80s, and John McClellan at University of Washington wrote a Cell paper in 2010 talking about strong evidence that suggests rare mutations of severe effect are responsible for a substantial portion of complex human disease. Now, this is a stark contrast to the dominant theme of polygenic risk score additive effect that we talked about, that the Boston group submits. So McLellan and King said this is not the model. The model is something like this, that the disorder results from any one of thousands of different mutations in any one of multiple different genes. So there are a handful of genes that are clearly known for breast cancer, BRCA1, BRCA2, etc. They're saying that there are thousands of different mutations within each of these genes that causes the disease with high penetrance, and collectively that is the genetic cause of the disease. They say genetic heterogeneity of inherited predisposition to breast cancer serves as a model for other complex illnesses. So they're not just talking about breast cancer, they're talking about many other complex human disease. And for us here, we are talking about breast cancer as an example, but we are interested in a variety of related effects, not necessarily breast cancer. So I'm going to go back to the initial data, puzzling initial data that I started with. So as you recall, monozygotic and dizygotic twins have the same lifetime risk, same number of years between the two breast cancers if they both have it in the twin. And conditional rate of breast cancer in monozygotic twin is about 1% per year if one twin had breast cancer, and that rate is half for dizygotic twins. But if the rate is half, why not the average number of years different? Why not twice or longer in dizygotic twins? That is a puzzling part. And we submit that the McLellan and King hypothesis explains this twin data. So if the co-twins carry a risk variant, monozygotic twin also carries a risk variant. That's 100%. Now, dizygotic twin have a 50% chance of carrying the same high risk variant because they are dizygotic. So this half of the rate makes sense. But how about the intervals? The key lies in the other 50%. In dizygotic twins, other 50% will get low risk because the high risk variant is not given to that person, the other twin. So it is unlikely both will develop breast cancer. So as far as both hard condition is concerned, the monozygotic twin and dizygotic twins are equal. So therefore, this is a picture that contrasts with the polygenic common variant model and the genetic heterogeneity rare variant model. So because it's smooth and continuous risk gradient created by PRS, dizygotic twin, if one has a high risk profile and then develop breast cancer, the other will be likely to be Toyota Corolla setting where half risk is shared of that high risk. On the other hand, McLellan and King hypothesis says, no, 50% of the time, monozygotic and dizygotic twins are the same. And then the other 50%, you don't get very much at all in terms of the risk. So therefore, this explains why the interval between the two cancers will be the same between monozygotic and dizygotic twins, which is inconsistent with this model. So if their hypothesis is the truth, why does PRS work? And that is given by our postdoc, William Letzel, who says common GWAS hits are poor surrogates for rare causal variants. So I'm going to explain this here using this picture. So suppose this purple is a rare variant that causes high risk, and the SNP in orange tags this haplotype. Then the orange risk is actually 1/45th of the true risk carried by this purple because only one person carries this and another 44 people who carry the same SNP tag but not carry the purple exist. So therefore, risk that is tagged by the SNP is only 1/45 of the real risk. So William's idea is because rare variant mutation must have occurred on a haplotype sometime, and therefore we need to find that haplotype. So suppose we are able to find that haplotype on which the mutation has occurred, then we should be able to go closer to the true risk. So in this illustration, this is tagged by SNP, so it's not a very good illustration, but green risk is only fifth of the true risk. So that is closer to the truth. How does he find it? He looks at chromosome overlap. He developed a method to look for shared allele patterns, possibly non-contiguous, in cases using a combinatorial algorithm and parallel computing. So he pairs the case chromosomes and find the shared pattern, and then find the shared pattern, and iterates. He applied that in three regions of GWAS hits, and I'm going to just talk about one of them, cyclin D1 region. He used the UK Biobank for discovery and drive oncoarray for replication. He found 3 rare haplotypes in this region from UK Biobank that carry the substantial risk, in particular this haplotype C. And when he looked at drive replication, 2 of the 3 haplotypes have replicated. Although odds ratio and hazard ratio are attenuated from the discovery, the third SNP carries a substantial risk, odds risk ratio of over tenfold by the haplotype. This region is a single TAD, topologically associating domains near the upstream of cyclin D1. And these are the SNPs that are shared by all 3 SNPs. And he found that a lot of transcription factor binding, especially CTCF, FOXA1, that is known to regulate differential interactions between ER and chromatin, both FOXA1 and CTCF. So there must be something here that could support the hypothesis and the finding of Williams. Also, the linkage study has reported a high load score in this region of chromosome 11, supporting some rare, highly penetrant genetic factor exists in this region. That is consistent with Williams' finding. Also, can we find rare risk haplotypes unrelated to GWAS hits? That is led by my team's FAM1. He looked for all the possible haplotypes with sliding window of certain SNP lengths. So he looked at 5, 10, etc., up to 500 SNP windows. He chops UK Biobank data for discovery before the enrollment of the study, and then used the prospective part after the enrollment for replication, and used the drive oncoarray data for validation. He looked at millions and millions of haplotypes and found 436 to be satisfying discovery and replication criteria. Now, if we do a permutation experiment, only about 10 haplotypes are discovered across these windows. So 436 is clearly beyond by chance. Of the 436, 193 are rare haplotypes, and they carry a high hazard ratio, risk ratio compared to the common haplotype. And looking at these 436 haplotypes, between replication and discovery, the hazard ratio estimates are quite consistent. Those are not representable by SNPs. If you talk about the common SNPs, common haplotype, you can represent a hazard ratio and p-value with the representative SNP in that area. But for rare haplotypes, hazard ratio and the p-values are both stronger for haplotypes than the SNP. So single SNP cannot represent these results. These are the 13 loci he found. I'm going to just go quickly because of the time. The hazard ratios are quite consistent between the discovery and replication. When he goes to the the drive validation study, only 6 of the 13 replicated, probably due to the fact that we have to impute in drive studies because of the different platform drive uses from the UK Biobank. One of the 6 areas is particularly intriguing because KIAA 1217, which has not been discussed for breast cancer at all, that protein is a gene is expressed most in female mammary tissue and also differentially by sex, according to GTEx portal. Also, Mayo Group has found fusion transcripts with SERPIN-A1 and KIAA1217 only in ER-positive breast cancer cases. And that's SERPIN-A1 is a target of estrogen receptor. So there's a binding site and expression of it in ER-positive cancer differentiates survival. Also, this gene regulates the transcription of ER-related genes, including FOXA1. So consistent with high-quality twin study, the hypothesis of McLellan and King are informing us how to look for genetic variants that are risk informative. Chromosome overlap and sliding window genome-wide haplotype analysis identifies rare haplotypes. And evidence from our 2 haplotype studies supports the hypothesis and explain why PRS works despite being inconsistent with the haplotype. So what did we learn? How we can utilize these for precision survivorship care and prevention. Our current conclusion is PRS adds to the prediction, certainly, and it is useful, but it should be improved with the hypothesis that is additional rare variants and haplotypes should be able to be found from the data that exist. We must mine the multivariate patterns and the survivor-specific PRS, including the gene treatment interaction, has to be developed. With that, I'd like to acknowledge my collaborators, in particular, Les Robinson, Yadav Sapkota, Xiaomi Wan, and Jinwei Zhang, and the Department of Epidemiology and Cancer Control, Computational Biology, Oncology, and Biostatistics at St. Jude, all the staff members who supports our studies. And in my direct team, Fan Wan, Wonjo Moon, William Retzo, and Cindy Im, and all the collaborators. And I'd like to thank my funding agencies. Thank you very much.
Well, thank you, Yutaka, for a great lecture. We have a few minutes left for questions, so I'll raise a few if you don't mind.
Yes.
Let's start with the McLellan-King hypothesis, which you were just teaching us about. Do you think that's entirely correct, that most of the risk of complex disease is really due to rare mutations?
So I think a lot of people would say that's a mix between the rare variant and the common variants. We need to see how that's going to turn out because the twin data is entirely consistent with the hypothesis. It doesn't look like the sum is come from the common variant. So I think we need to investigate further, but I believe that some of the diseases must be genetic heterogeneity with a lot of rare mutations or haplotypes.
Okay. Well, here's another Yutaka. When assessing genome-wide genetics, there's always the challenge of false discovery. If a source for replication is not available, what other approaches might be used to support results from discovery-based analyses?
That is a difficult question. It has to be biological. Statistics and epidemiology uses data replication as a way to validate the findings. but eventually biologists must discover why this haplotype is associated with the disease functionally and otherwise, like Mendelian disease.
All right, great. We have another question from Kim Nichols. Great talk, Yutaka. Will you be looking into SNPs as modifiers of disease penetrance conferred by high-risk autosomal dominant gene mutations?
Yeah, that is that there has been a report for those. I think we need to carefully examine those. That is a possibility. So gene-gene interaction, SNP-SNP interaction, one gene modifying the other gene's effect, that's highly probable, and that is an area of high interest.
Okay, good. So, what obstacles do you see for applying genetics within pediatric cancer survivors to assess interactions between treatment exposures and genetics? What are the obstacles or the barriers to really doing that well?
So we are digging into genetics because there's no good sort of template that we can apply directly to the cancer survivors. So we have to go into the genetics, dig deeper, and trying to develop methods. And that's what Cindy Im and Yang Yuan are doing. And also in survivorship research, although the survivors numbers are increasing, numbers are still small. So for rare mutations, rare haplotypes, it is difficult to study that directly. And also St. Jude, CCSS and St. Jude Life are very good resource, but we need more population to collect genome information so that we can use them for replication and confirmational study.
Yeah, and maybe one more follow-up question before we close, and that is, this idea of interaction is so important. And you mentioned that Cindy M will be looking at survivor-specific polygenic risk scores. I assume it's by leveraging those interactions. Maybe you could go into a little more detail on the methods of that and how that will be unique, it sounds.
So the idea is that the treatment exposure definitely dictates the risk of the late effects. And we know that anthracycline develops cardiomyopathy, but not everybody who gets 250 milligram per square meter anthracycline develops cardiomyopathy. Then that's where the genetics comes in. So interaction of background genetics and exposure is important. But not everybody is exposed to anthracycline. So the information is limited among the anthracycline exposed population, and cardiomyopathy occurs much later than time of the diagnosis. So it is a challenge, but that is what we are trying to address and needs to be addressed specifically for survivor population.
Well, thank you, Yutaka. We are at the top of the hour, so I'd like to thank you for a wonderful talk. At this point, we'll close the official event, and then we'll invite all who are interested to stay on for questions to stay on with us. We'll have an extended Q&A immediately following this. I want to thank all of you for coming. Please join us next Thursday at the same time when Dr. Suzanne Baker of St. Jude will be presenting Disrupted Development in Pediatric High-Grade Glioma. Also, mark your calendars for another exciting survivorship talk on May 12th when Dr. Lindsay Morton of the National Cancer Institute will present on genetic susceptibility to subsequent neoplasms following childhood cancer. So thank you again, Yutaka. This
Thank
closes
you.
the official event, but let's go into some extended Q&A. We have some other questions here if you're ready for them.
Yes, thank
Um,
you.
we have one from Evandy Rampersad. Nice talk, Yutaka. Can you speak to the utility, accuracy, and predictive capability of PRS in cohorts with considerably smaller size?
I think the accuracy will be dependent on the sample that you obtained. So is it unbiased sample, representative sample, or hospital-based, convenient sample? Accuracy, precision will be dependent on the size of the population and the outcome frequency. So I think you cannot generalize necessarily, but I think if like Cindy's research showed that if there's no other external factors that dictate the risk of the phenotype, then you expect the general population's PRS to be applicable to a specific small group of patients or some other characteristics of the people.
All right, thank you. Here's another one. You talked about mining multivariate patterns. And so exactly what types of patterns do you mean and how would you go about doing that?
So that relates to this one. So for example, if you look at this fund's statistics, I didn't go through it carefully, but he tested millions of haplotypes. But effective window size is only 650,000, which is the number of SNPs that are genotyped in UK Biobank. So that means, let's say for window 50, 88 million haplotypes in the 645,000 windows. That means for each window, there are more than 100 haplotypes. That frequency of more than 0.1%, that's what Fan tested. And if you go to rarer haplotype, even more than the 88 million. So there are lots and lots of haplotypes within the same window. Now we are testing those according to, sorry, I have to go to another screen. That is what I talked about here. So here I talked about five haplotypes, and if you look at one SNP at a time, you're comparing one versus all the rest. Imagine you have hundreds haplotypes in a window. Then you're testing 1 versus 99, and each time you're just comparing one versus others. Now, if one of the 100 is relevant for the risk, that's fine. But if it's more than one, because they're in the same locus, same region of the chromosome, why one, unless it is a rare mutations, why one is relevant for the risk? So we have to look at those together and trying to find each haplotype's risk simultaneously. That's what I mean by multivariate.
Great. Excellent explanation. Thank you, Yutaka. So Jun Yang has a question for you. Have you explored Mendelian randomization to determine the causal effects of specific therapeutic exposures on late effects using genomics data that you have already in hand?
I personally haven't, but I think some of the interest exists in biostatistics department, I think, and also Yadav Sapkota has expertise on this. I think Gorian in biostatistics department and her postdoc looking at Mendelian randomization.
Okay, sounds good. And then, you only found a small number of rare haplotypes associated with breast cancer risk. So do those really explain the whole risk captured by GWAS hits, or is there more?
Yeah, that issue is so we calculated power for this analysis. So in order to pass these three steps in Fan's analysis, discovery, replication, and validation, we have very limited power. And so that means we found, let's say for this one, we have power of 2.3%. So what we observed is one of the 2.3% that we found here in this analysis. So there should be 43 more that are similar disk size and frequency. So if you follow that logic, we have many more that we couldn't detect because of the low power, and we could detect if we had high power study. Also, we use only 650,000 SNPs that are genotyped in UK Biobank. So if you had the whole genome sequencing data, which UK Biobank is providing soon, we should be able to test many more haplotypes. And therefore, these two together, we are missing a lot of haplotypes. And so although we have very small number of discovery, I think this might explain why we only find a few, and there might be many more that could replace the GWAS hit risk patterns.
Great. So Ching raises the question, I wonder if it makes sense to borrow strength across rare haplotypes to increase power. Is that possible?
Yes, certainly. So the current rare variant analysis idea, such as SCAT, etc., combines different rare variants. So certainly we can combine rare haplotypes and test it. We haven't done that. If you are interested, let's collaborate. I would love to see how that works.
Great. And Yutaka, you are an international expert in cohort methodology. Your 20 years of plus of CCSS, St. Jude Life. I'd love to pull back and ask you some larger cohort methodology questions, if you don't mind.
Okay. Yeah.
You know, the one of the things that comes up often is the idea of survivor bias, and a lot of times that is ascribed when there's really non-survivor bias. There are actually situations where there's real survivor bias, and it's problematic. I'd love to let you just talk about survivor bias. What is it really? What is and isn't survivor bias, and when is it problematic?
I think we have to be careful talking about survivor bias because here we are talking about our target population is survivors. So if we collect all the 5-year survivors without any selection, then that is an unbiased, cohort of 5-year survivors, and there's no problem. That is not a bias because we are getting the target population entirely, and it's not a biased sample. Now, typically, survivor bias talks about if you survive, you get into the study. Now, that is a bias, and that needs to be addressed. And in order to address that, you need to have information about the original target population and sampled population, and then you compare. And if you have a solid information between the two, you can kind of make assumptions about representativeness stratified by the characteristics and do inverse probability weighting or some other techniques to address the target population's association rather than just the survivors. So we have to distinguish these two types of survivors. Because we are studying survivors, we cannot say we are always subject to survivor bias. So it is important to consider these two separately, and especially CCSS, which collects all the 5-year survivors, although participation is not 100%, but next of kin is providing information for the deceased survivors that at least try to address that survivor bias issue.
Right, and maybe you could take a deeper dive with that and some of the genetic studies, particularly second cancers, where very bad subsequent neoplasms, so survivors may have died earlier than later. Are we seeing a tremendous amount of bias in some of these studies due to that?
We don't know because we don't have the genotype of the second cancer survivors who passed away before the study took place. So if the sample is collected from the survivors, so it's a prospective collection of the data, and the blood sample or DNA was not collected at the time of diagnosis before they become 5-year survivors or something like that, then we might have the survivor bias if you collect the people from the people who are alive today and not from the people who have passed away.
Good, thank you, Tyra. And now another area is cumulative burden. This is a relatively new methodology that you developed, and necessity is often the mother of invention. And then this is one of those cases, I think. Maybe if you could tell us a little bit about why there was a need, maybe I guess a decade ago, to really think about cumulative burden, and then how you came about really quantifying that correctly, and then what advantage it gives us over cumulative incidence and other approaches.
So this is not I just did the statistics. And the original idea was from Les Robison. I remember in Minneapolis when we were in a meeting, he called me to a cafe and told me, Yutaka, there's a issue in survivorship research. That is, as survivor age, there's more and more conditions they develop, chronic health conditions because of aging. And how do we quantify that? Can't we quantify those multiple conditions occurrences, which cannot be captured by cumulative incidence, which is for the first event only? And when I heard that, we worked on statistical methodology, and then Nikhil took much higher level, considering clinical issues of different chronic health conditions and how to add them up, or is it permanent conditions versus temporary conditions? Those things are all sort of in the methodology of cumulative burden. So we are basically adding up the cumulative incidence of different conditions, and together we are trying to address and describe how many multiple conditions over time survivors develop by their different characteristics.
Right, and it's really become a critical metric in the field that we're seeing many, many other study groups using. So thanks to you, Nikhil and Les. Well, Yutaka, that is the last of the questions that I see. So I want to
Right.
thank you again for a great lecture and
Thank you.
for staying on for the extra period to answer these questions. And then again, encourage everyone to join us next week for Dr. Susie Baker, who will be giving the talk next week. All right, have a great day, everyone. Thank you for
Thank
joining.
you.
All
Thank
right,
you.
bye-bye.
Bye.